Hi, my name is Rami Mehio. I am VP of Bioinformatics and Instrument Software at Illumina. I'm here to talk to you about running genomic workloads using Illumina's DRAGEN on AWS. This is work that we've been doing at Illumina with AWS since 2016. The Illumina acquired DRAGEN, a company called Edico Genome, in uh, 2018. The work started on running DRAGEN on AWS in 2016 with a lot of collaboration. And I'm going to present to you the evolution of that and where it stands. So first, a few words on Illumina. Our mission is to improve the human health by unblocking the power of the genome. So Illumina is a sequencing company. What it does really is uh, takes your blood samples or saliva and extracts DNA and sequences it to see what are the mutations and variants in your genome that determine your health risks, determine your predisposition to certain diseases, and so on and so forth. And our mission is to help the world take advantage of that knowledge to improve human health. So, how does that work? The DNA of each person is in the chromosomes, and it's made of two strands. The bases are A, C, T, and G. And the sequencer that Illumina builds, or the sequencers that it's been building for many years now, and is responsible for the vast majority of sequencing that has happened of humans in the world, is able to detect from your blood or saliva what are those bases in your DNA. And we do that regularly. We're able to sequence, hundreds of genomes per day on a machine. And it's used for both research and diagnostics. And beyond the sequencing step, what's really important is there is an analysis of the DNA that has to happen. And that analysis is done through informatics and software, and as you can see, sometimes hardware and hardware acceleration that is meant to basically bring meaning to the sequenced DNA. So just to put this into context, the number of genomes sequenced per year, as you see with the green curve here, has risen very significantly over the past 10 years. And that coincides to a large degree with the drop in price in the cost of a genome. The first genome cost, north of $100 million to basically sequence. And over time, Illumina, with the acquisition of Solexa around 2007, 2008, was able to start the trend of dropping the price very significantly. And of course, along with the cost of sequencing, the accuracy and the reliability of sequencing has come along. So as you see, there are a lot of genomes being sequenced now, and that brings power to this genomic industry that's able to make use of the sequencing to discover drugs, to find pre-existing conditions and diagnose disease. So an example of a great foray in this diagnostic world is the recently announced acquisition by Illumina of Grain. It's a company based in the Bay Area that uses our sequencing technology of DNA to look at blood samples to screen people for cancer. It's able, by looking at the DNA and the methylation around the DNA, to identify or detect early signs of cancer for up to 50 different cancers. So you could see the sequencing technology is a key enabler to improving human health, and that's a very important example of it. That's only possible now as the cost of the sequencing has dropped down significantly, and of course the number of use cases for sequencing and the number of genome analyzed has gone very high. Now, the emergence of sequencing as a very important aspect of human health or improving human health, and the amount of sequencing happening and the number of genomes going through the machines comes with a certain computational problem. Each genome has about 3 billion bases, these A, C, G, and T. And when they are sequenced and the output needs to be analyzed, it requires a lot of horsepower to be able to extract the meaning and detect the variance in that genome. And over time, it used to take, and if you see in the graph on the left, how much, the amount of hours it takes to sequence every genome or to analyze every genome, and how much effort people are trying to put to reduce that, but it still adds up to, multiple hours per steps. And we're talking here about small sequences, and these are graphs from a few years ago showing Intel's effort in this area to quantify it, and they've done a lot of work in this area. So each genome takes a huge amount of a compute, either a lot of hours or a lot of cores or a mix of both. And of course, writing Moore's law helps us essentially tackle this problem. So with the advancement in CPUs, the advancement in the compute capability of all the new CPUs and multiprocessors, we're able to shrink that time. But also, on the other hand, our sequencers used to produce half a genome or a quarter genome every run, every 24 hours. Now these sequencers produce 48. You can imagine in the future much more. And there are many of them around the world, showing a lot of usage and producing many genomes. So that puts a significant strain on the compute resources. It takes a long time to analyze, but also Moore's law is showing signs of flattening. And you see here around 2010, it becomes harder and harder to get higher clock speeds. It's harder and harder to get bigger chips. So Moore's law keeps evolving, but significant obstacles are there to keep up with all the genomic analysis, given how much computationally intensive those are. So this is where DRAGEN and the technology that Illumina has acquired from Adico comes. So you could see the world actually moved in two directions to solve this problem, and both very interesting. On the left-hand side, you have this move to use the cloud and the power of the cloud and divide the genome to be analyzed into multiple small pieces and spread it across as many instances as possible, this reduces the amount of time it takes to analyze the genome, but doesn't solve the need to spend a lot of cores and compute hours in total to achieve the task. On the right-hand side, there's the other class of solution for this problem, which is to accelerate that analysis through non-traditional CPU means. And in that space, you have the class of accelerators such as GPUs, FPGAs, and other accelerators, perhaps ASICs, application-specific integrated circuits that can come to play. Illumina and Edico Genome chose the path of accelerating the analysis through FPGA processors. Those are off-the-shelf chips that are configurable, that can make circuits out of low-level logic, such as AND gates, OR gates, memories, multipliers, and they are could be programmed to build circuits that achieve specific tasks in the most streamlined way. So in 2016, AWS introduced the F1 instances in 3 flavors, the 2x, 4x, and 8x. We talk today here on the 4x, one of the instances. It's an 8-core, so compared to the latest C5 and C6 instances, it's very small in terms of cores, but it has an FPGA card, which is a card that has this FPGA chip and some memory, very similar to GPU cards, but treats the acceleration in a different way. With that, the Edico solution from Illumina, DRAGEN, is able to analyze the genome in 30 minutes with 8 cores and 1 FPGA, compared to hundreds and hundreds of core hours to do the same in the traditional means. But also the difference is quite fundamental. It is you're relying on a single machine to execute your job. And the other side is you're relying on many machines or many cores to do the work in parallel. So the logistics and ease are very different. On the right-hand side, it's much more reliable operation. It's a single machine. And from a cost perspective, even if you're using on-demand processing, it gets you a really low cost per genome. And if you add to that the ability to do spot processing on AWS, that number will go significantly lower. So let me speak a little bit more about the solution. So this solution really is a server, an instance on AWS in which there is an FPGA card. You see it here. It's one example of those. Of course, that solution could be present on site as well. There is no, Illumina does provide those on site for customers who cannot connect to the cloud. And the chips are off-the-shelf chips today that we use from Xilinx. There are other vendors like Altera and others that have also FPGA chips that could be used for the same. With that capability, the hardware, the FPGA, and the chips, Illumina and Edico have built a software stack that not only analyzes the genome for one use case, but it is able to analyze it for applications of inherited disease, of copy number variations, of cancer mutation detection, of biomarker discovery, of RNA, of methylation, and other. So we have built a whole class of analysis tools on top of this hardware technology that could be run on site and on the cloud. and we offer it to be run as an Amazon machine image on AWS on these F1 instances. It could be Dockerized or not. And we also offer it behind our SaaS service, be it BaseSpace or Bluebee that we acquired recently. But we've also offered it in other platforms as well, such as DNA Nexus and the 7 Bridges. So how does that work? In general, the solution is the software on the left, the FPGA board on the right, and so it's all these pipelines and the analysis solutions are abstracted from the hardware through hardware abstraction layer HAL. And the way it works is the software would start computing the analysis of the genome, and when it encounters an operation that is very expensive to compute on CPUs, it will send it over the PCIe bus to these FPGA boards, and the FPGA boards would be loaded with these circuits that are pre-designed to handle these highly complex and very computational intensive operations, and return the answer to the software to continue. And as each analysis solution asks for a specific type of computation, it gets loaded from a library of pre-built engines. So as you see on the right, we have DNA mapping, RNA, Smith-Waterman, graph, zip, unzip. And we have a whole suite of those for analyzing genomes, but we also have them for machine learning. So the architecture you're seeing here is generic. It is not specific to genomics. However, we have made great libraries for genomic application. We are venturing and adding capabilities in spaces of machine learning, compression, and image recognition that we keep adding to the platform to complete our solution. So generalized architecture with a huge vertical in genomic analysis that we have packaged as applications to be used on these F1 instances and on our on-site solutions. Now, with such tremendous compute, and, this compute that cuts analysis from 20 hours to 30 minutes or 20 minutes, you get also the luxury of investing in algorithms that otherwise you would shy away from. You shy away from because they are very costly to compute. It takes too much time. It will cost too much to pay for the instances. So people end up cutting corners on what's the best possible algorithm they can implement to detect variants and diagnose disease and so on and so forth. So one of these key examples that Illumina has now been able to capitalize on is a recent challenge, the Precision FDA challenge organized by the Precision FDA and NIST. This is a consortium for the genome in a bottle where they produce truth sets about genomes, and they've organized a competition to evaluate the best tools to detect variants and analyze genomes coming out from sequences. And Illumina and another 20 participants from the industry, such as Google and Roche and others, and they're listed here on the bottom, participated to analyze the genome out of the Illumina sequencers. And we fully took advantage of our compute capabilities with these FPGAs and the F1 instances to make the best algorithm. And the graphs that you see on the bottom show the number of errors each one of the tools has made when it analyzed the genome. And as you can see, DRAGEN has about between 28% and 38% lead over the next best tool that does it. And that's to a large extent to the superior algorithm that we are able to take advantage of, both from a design, but also from the ability to compute them efficiently in FPGA. So the combination of both the sequencing and the right software and the compute capability puts the solution in a very special place for doctors and researchers to get the best information out of the money they spent on a genome, but also doctors to diagnose disease from their patients. So let's take this a little bit to a real life example. So the Broad Institute and Illumina have a very long-lasting and evolving collaboration. More recently, in the past couple of years, on bioinformatics between the DRAGEN team and the GATK team, and working together. This is a poster from the Broad Institute on how they run their genomic analysis at the Broad Institute. They have one of the biggest sequencing labs in the world, and one of the most respected. And so today they run the analysis through the DRAGEN pipelines on site, and in an HPC type manner with Slurm. And they have a fleet of sequences on the left, putting data into storage. They get analyzed and sent through through a fast interconnect, through a scheduler to the different compute nodes that are DRAGEN, and they're able to produce genomes every minute or every few minutes through that, which is typically an operation that would take 20 hours or so. So they've established that solution as an HPC cluster of DRAGENs. And of course, as their demand goes, they need to be able to scale up or scale out to the cloud. And this is where the such labs can take advantage of also the cloud solution. So with that, we have many of those customers, and a lot of them either start on site and move to the cloud, or they have they're starting on the cloud straight, but also sometimes they have a hybrid solution. And the HPC capability of AWS, the parallel cluster, the elastic fabric adapter with the high-speed interconnect, and the Lustre storage is ideally suited. We at Illumina have such a setup on site, and we are in the process of looking how to scale that out. The fact that F1 instances are present allows us to basically do just that. So you could imagine setting up a parallel cluster of F1 instances, which have built-in very fast hard disks, have the FPGA, the DRAGEN application connected to very fast storage and network at a high speed. And you can imagine the fleet of sequencers producing fast queues, which are the input files to the analysis, and transferred to the cloud for analysis, or done in a hybrid way. What now complements this solution very nicely is also Illumina's acquisition of Enancio, which is a compressing technology, allowing us to compress these input samples by a factor of 5, making the transfer much easier. These files are typically for a whole genome about 50 or 60 gigabytes. With the compression technology in DRAGEN now that acquired from Enancio, these files go down to about 10 or 12 gigabytes, making them much easier to handle to send to the cloud, cheaper for your storage, but also allowing you for an efficient scale-out operation where if you have your system on site, you can scale out and you can move the data in a much easier way with the compression available. So this is how you would set up your production for analysis. But once you are on the cloud, that also opens the gate for moving past that. If you have your setup through HPC or through Amazon Batch, a typical deployment, either way, that opens up to adding a layer of applications, meaning above this HPC cluster, you could have a hosted endpoint and a website where you can offer the different analysis that DRAGEN does. So you can see the methylation, the somatic, the RNA, the liquid biopsy, many other examples available with DRAGEN. It's really pretty simple to go from the basic DRAGEN application that Illumina offers to building this whole infrastructure and having a solution that can encompass many applications, user interface, website, down to the analysis in a very, very fast speed. So let's step back a little bit. We started really, 5 or 7 or 8 years ago with a difficult problem, which is how can we analyze quickly, and how do we not go through hundreds of nodes or many hours of compute. With the help of with the design that DRAGEN did with the FPGA, with the deployment of F1 instances by AWS, by having services such as AWS Parallel Cluster, and the ease at which you can build a website on top of that, you can go really from a very difficult computational problem to an easily buildable app store for application that you can use for your genomic analysis. So I'd like to close with that. Thank you very much, and thank you.